Welcome children. In our last class we discussed about the structure of DNA and composition of DNA and how the polynucleotide chains are formed, what is nucleotide, what are the composition of nucleotide. So today we will be discussing these things children. We will be discussing the contribution of scientists related to structure and composition of DNA, under which we will discuss about the contribution of Friedrich Michel, contribution of Erwin Chargaff, and the major contribution by Watson and Crick. Okay, then we will discuss packaging of DNA. तो चलिए बच्चों, सबसे पहले हम डिस्कस शुरू करते हैं फ्रेडरिक मिशेल का क्या कंट्रीब्यूशन था. सो प्लीज ऑल ऑफ यू नोट डाउन दिस. विल डिस्कस वन बाय वन. So the first contribution was made by Friedrich Michel. He said that, let me write this. He said the nucleic acids are the acidic substance found in the nucleus. Okay, so that's why they are known as nucleic acid, and as you know the nucleic acids are of two types, that is RNA and DNA. Okay, so they are first identified by F. Michel in nucleus and they were acidic in nature. That's why they are known as nucleic acid. So this was the contribution of Friedrich Michel. Now what is the contribution made by Erwin Chargaff? Okay, so next one is contribution by Erwin Chargaff. So second one is Erwin Chargaff. Okay, so Erwin Chargaff, he proposed a base pair rule. So he proposed a base pair rule after studying the base pair nature in different species. बहुत सारे स्पेसीज में उन्होंने एक्सपेरिमेंट किया and then he got the conclusion. What is that conclusion? That conclusion was that the ratio between adenine, that is A, and guanine, I'm sorry, the ratio between adenine and thymine and guanine, that is G, and cytosine is always constant. Okay, that means A plus T and G plus C, ये दोनों का रेशियो कितना होगा? The ratio will always be 1. Okay, on the basis of this rule, children, you can do many numericals. So here I will be discussing only one numerical, and after understanding that numerical, you can solve all other kind of numericals related to this. Okay, so note down the numericals. If a double-stranded DNA has, if a double-stranded DNA has 20%, suppose say cytosine, 20% cytosine, find out the out the percent of adenine present in it. Okay, so how to solve this? See children, cytosine, that means C is equal to 20%, and we know that according to Chargaff's base pair rule, the number of C is equivalent to number of G, because always there is a constant pairing between C and G. C जितना रहेगा G उतना ही रहेगा. That means this implies that G is also equal to 20%. So what is C plus G? So C plus G is equal to 20% plus 20% is equal to 40%. Okay. And what about A and T? So now what is remaining? A plus T. What will be A plus T? A plus T is equal to 100 minus C plus G. That will be equivalent to 100 minus 40, that is equal to 60%. And we know that the number of A is equal to number of T. Okay. So, if A plus T is equal to 60%, that means A is equal to T is equal to 60 by 2 is equal to 30%. Okay. So, what is the amount of, that is present your adenine or thymine? It is 30%. Okay. Now, in this way, you can find out the constituent or the base present in the DNA in applying this rule or applying this formula. Okay. Now, let us discuss the contribution made by Watson and Crick. Okay. So, here I will write, in last class I have already explained the points, and that is structurally I have explained the double helical model proposed by Watson and Crick, and today I will be discussing the Watson and Crick model of double helical DNA. Okay. So, इसका क्या क्या उन्होंने DNA के बारे में बताया? The DNA is made up of two nucleotide chain in which the backbone is made up of phosphate and sugar, and the nitrogenous bases are projected inside. What does it mean? Suppose say this is a DNA strand, and in the DNA strand, as I have said you, it is a polynucleotide chain. So this is the structure of nucleotide which are arranged like this in a polynucleotide chain, and this side the base is present and this side phosphate is present. So this is phosphate and this is sugar. So the sugar and phosphate forms the backbone of the DNA strand, whereas the base pair is projected inward. Similarly, in the other strand, the base is facing this way, and and the sugar and phosphate forms the backbone. Okay. Now next point. The two polynucleotide chains, the two strands of DNA are anti-parallel to each other. Means if one strand is in 5' to 3' direction, then the other is in 3' to 5' direction. What does it mean? ये देखो दो स्ट्रैंड है डीएनए का. ओके, अगर ये स्ट्रैंड 5' टू 3' डायरेक्शन में है तो दूसरा स्ट्रैंड 3' टू 5' डायरेक्शन में होता है. अगर ये ऐसा है तो ये अपोजिट डायरेक्शन में जाता है. एंड आई हैव ऑलरेडी टोल्ड यू व्हाट इज 5' एंड व्हाट इज 3'. ओके, नाउ द नेक्स्ट पॉइंट. The third point is that the base pair are formed by the base pair is formed by the pairing of nitrogenous base by हाइड्रोजन बॉन्ड. हम आपको बताए थे A pairs with T by 2 हाइड्रोजन बॉन्ड, G pairs with C by 3 हाइड्रोजन बॉन्ड. ओके, सो हियर A pairs with T by 2 hydrogen bond, G pairs with C by 3 hydrogen bond. Okay, and there is always pairing between purine and pyrimidine. Hence, a uniform diameter of 20 Armstrong is maintained. In a DNA molecule. In a DNA molecule. You know that, children, purines are single ringed structure. Okay, they are single rings. Purines are double ringed structure, and pyrimidines are single ringed structure. So when there is a constant pairing between double ring and single ring structure, then the uniform diameter is maintained. अगर T के साथ प्यूरिन के साथ प्यूरिन होता तो क्या होता, वहां पर डायमीटर ज्यादा हो जाता. अगर पिरिमिडिन के साथ पिरिमिडिन का बॉन्डिंग होता तो क्या होता है, वहां पर डायमीटर कम हो जाता. क्योंकि यहाँ पर हमेशा प्यूरिन के साथ पिरिमिडिन का जो बॉन्डिंग है वो कांस्टेंट रहता है. दैट्स व्हाई the diameter of 20 Armstrong, it is not exactly 20 Armstrong, it is approximately 20 Armstrong is maintained. Okay, now let us discuss the next point. The next point is the two strands are coiled in right-handed fashion. Okay, it is right-handed fashion, and its pitch is 3.4 nanometer or 34 angstrom. Okay, so children, you know that there are different types of different forms of DNA: A form, B form, D form, E form. So in some cases the DNA is left-handed, in some cases it is right-handed, in some cases it is zigzag. But in most of the cases the DNA is B form, and the double helical structure that was proposed by Watson and Crick was a right-handed DNA. That was the B form DNA. So the pitch is 3.4 nanometer or 34 angstrom, and each turn consists of about 10 base pairs. So the distance between two successive base pair will be how much? It will be 34 angstrom by 10 or 3.4 nanometer by 10 is equal to 0.34 nanometer. Okay, so in this way we can find out the distance as well as the length of DNA molecule. One more thing, the fifth point is that the DNA is stable, more stable. The stability of DNA is due to the stacking of base pairs and also due to the hydrogen bond. ये देखो दो स्ट्रैंड के बीच जो बेस होते हैं उनके बीच हाइड्रोजन बॉन्ड होता है. दिस इज हाइड्रोजन बॉन्ड. So this hydrogen bond provides extra stability, and besides that you can see that the base pairs are stacking over one another. एक के ऊपर दूसरा पेयर ऐसे arranged है. Okay, and this provides extra stability to the DNA molecule. Okay, so our next topic is children, I think the contribution of scientists and structure of DNA, composition of DNA, everything are clear to you. Now I will discuss the packaging of DNA. Packaging of DNA. Children, you will be wanted to know that such a big DNA is how it is accommodated in such a small nucleus. And in the last class, I have told you about the length of the human DNA. And the human DNA or any DNA, the length of any DNA is measured in terms of base pairs. So length of DNA is measured in terms of base pairs. So the total number of base pairs multiplied with the distance between the two consecutive base pair gives the length of the DNA. Okay, so I have told you that in the haploid set of chromosome, that is in 23 chromosome, what is the length of what is the number of base pair? The number of base pair is 3.3 into 10 to the power 9 base pair. Okay, but in the whole set, I mean the diploid set of chromosome, this is in haploid set. Haploid set of chromosome in human being, but in diploid set of chromosome, it is how much? In diploid set, it will be 6.6 into 10 to the power 9 base pair. So this is in diploid set of chromosome, that is in total altogether in 46 chromosome. Okay, now how to find out the length of the DNA? इसमें DNA का length हम कैसे find out करेंगे? Okay, so here the length of DNA in diploid set of chromosome in human being will be, as I have told you, the number of base pair multiplied by the distance between consecutive base pair. Okay, so what is the base pair, number of base pair? 6.6 into 10 to the power 9 multiplied by what is the distance between consecutive base pair? It is 0.34 nanometer, and we have to convert nanometer into meter. Okay, so multiplied by 10 to the power minus 9. So this 10 to the power minus 9 we are multiplying in order to make it in meter. So it will come around 2.2 meter. So this is meter per base pair. So it will be around 2.2 meter. Okay. So now see, it is around, it is not exactly, approximately. Okay. So see, such a long 2.2 meter DNA, how it is accommodated in our small nucleus, which is even cannot be seen clearly under compound microscope. So it is just strange. So now it is very important to know how such a big DNA molecule is packaged in such a small nucleus. Okay. So now we will be discussing the packaging of DNA in prokaryote as well as in eukaryote. Okay. So let us start with packaging of DNA in prokaryote. As you know, in prokaryote, a single DNA is present, single looped DNA is present, and there is no nuclear membrane, definite nucleus is absent. So in case of prokaryote, first one is in prokaryote, how the DNA is packaged? The DNA in prokaryote means in bacteria, they do not have nucleus, and but still, suppose this is a bacterial cell, they don't have nucleus, but it doesn't mean that the DNA is moving here and there. A single DNA is present, looped DNA, but it doesn't mean that the DNA will move here and there. So it is confined to a particular region, and that region is known as nucleoid. That region is known as nucleoid. Okay, and this is how the DNA is confined to a particular position. It is because of the packaging of the DNA, and that packaging is possible due to the negative charge of DNA and positively charged protein. Okay, तो देखो बच्चों यहाँ पर क्या होता है in case of prokaryote, nucleus is absent but looped DNA is packaged in a confined region called nucleoid. DNA is negatively charged and it is forming a loop with the help of a positively charged protein. Okay, so this is how the packaging of DNA in case of prokaryote is very simple. A negatively charged DNA is negatively charged and it is packaged with the positively charged protein. That's why it forms a looped structure and it is confined to a particular region that is called nucleoid. This is nucleoid. Okay. Now, how the packaging of DNA takes place in eukaryote? But because in eukaryote so many chromosomes are there, so many DNA are there, so the length of DNA is much more than that of the bacteria or prokaryotes. Then how this packaging is taking place? So let us discuss that one. Now, packaging of DNA in eukaryote. In eukaryote. Okay. So, in eukaryote, a set of positively charged protein in eukaryote, a set of positively charged basic proteins, the proteins which helps in packaging of the DNA are positively charged as well as they are basic in nature and basic proteins and called histones. Very important, children. Histones are present. This protein, histone, is positively charged due to presence of two amino acids. So these two amino acids are very important, children. You have to remember, that is lysine and arginine. Okay, so presence of lysine and arginine in large number. Okay, so the protein is positively charged and these proteins, this is a set of protein and it forms a histone octamer. It forms a histone octamer. Okay, so the proteins form a histone octamer. That means it composed of 8 subunits. 8 subunits or 8 molecules of protein That's why it is known as histone octamer, which is positively charged. And I am not talking in details about the histones. Actually, histones are of 5 types, like H2A, H2B, H3 and H4, and they are but here we are discussing about the histone octamer, which is having H1 unit as well. So this histone is positively charged, whereas the DNA is negatively charged. Now there is a tight bonding between this positively charged protein and negatively charged DNA. Okay, and they form a unit. They form a unit that is called nucleosome. So the packaging unit of DNA in eukaryote is called nucleosome. So we'll discuss about nucleosome. Nucleosome is the packaging unit of DNA in eukaryote. So this packaging unit is formed when the negatively charged DNA wrap around. So this is formed when a negatively charged DNA is wrapped around the positively charged histone octamer. ठीक है, ये ऐसे रैप हो जाता है. मान लीजिए हिस्टोन ऑक्टामर एक बॉल है और इसके चारों तरफ डीएनए ऐसे रैप हो जाता है, and it forms a unit that is called nucleosome. And a nucleosome contains a single nucleosome. So एक क्रोमोसोम में एक लंबा सा डीएनए होता है और वो डीएनए ऐसे कॉइल ऐसे होता है. ठीक है, सो ये एक न्यूक्लियोसोम होता है. दिस इज ए nucleosome, and each nucleosome contains 200 base pair approximately. ठीक है, so what is the number of base pair in the nucleosome? It is 200 base pair of the DNA helix. Okay, and this nucleosome forms a repeating unit as I am showing here. यहाँ पर आपका जैसे मैं दिखा रही हूँ, it forms the repeating unit. So this is a nucleosome. This is a nucleosome. ये ऐसे repeating unit बनाता है, and in this way such a long DNA is packaged in the nucleus. Okay, and it forms a coiled thread like structure. जैसे कि एक thread में beads are there in the string में beads है, उसी तरह arrangement इसका होता है. But the single packaging unit of DNA is known as nucleosome. Okay, so this can be visible under electron microscope only. So now let me draw the structure of a nucleosome. How a single nucleosome look like? This diagram is very important, children, from the examination point of view. ठीक है, ये डायग्राम आप जरूर प्रैक्टिस करना, और ये पैकेजिंग यूनिट कैसे बनता है दैट ऑल्सो यू हैव टू प्रैक्टिस. ओके, सो दिस इज द हिस्टोन प्रोटीन. ये हिस्टोन ऑक्टामर है, एंड दिस इज H1 हिस्टोन. दिस इज H1 हिस्टोन. और इसके चारों तरफ क्या होता है बच्चों? द DNA इज wrapped around. I'm not so good in diagram, but still I'm trying to do the good diagram. Okay. The next is your okay. So this is the whole structure is the nucleosome. Here this is H1 histone and ये सब यूनिट जो है this is H1 histone and this is only this rounded structure is histone octamer, okay, and this is the DNA and this DNA is of 200 base pair. Now you can see this is the part of DNA that is projecting out. Now it will come and again it will form another nucleosome. So this portion of DNA which is not forming the nucleosome is known as linked DNA. Linked DNA. Okay. So the whole structure is nucleosome. This is the packaging unit of DNA. How is it possible? It is possible due to the positively charged histone protein. Why histone protein is positively charged? Because it is rich in positively charged amino acid, that is lysine and arginine, and it is basic in nature, whereas DNA is negatively charged. So there is a strong bonding between positively charged protein and negatively charged DNA, and the DNA wraps around the histone protein and it forms a packaging unit that is called nucleosome. Okay, so this is how the packaging is taking place, and these units are arranged in such a way to form a structure that is called chromatin fiber, but besides the histone protein there is another protein that is called non-histone chromosomal protein. So I am rubbing this one. So that is NHC, non-histone chromosomal protein. So for high level packaging, non-histone NHC, very important children, non-histone chromosomal protein. Okay, so this protein is another positively charged additional set of protein that are required for the packaging of chromatin at a higher level. This is required for higher level packaging. Okay, so that is beyond your syllabus. Now children, one more point I will tell you. Depending upon the packaging, how the packaging units or nucleosomes are arranged in a chromatin fiber, depending on that, the chromatin fibers are of two types, that is euchromatin and heterochromatin. Okay, so euchromatin and heterochromatin. So let me first explain you diagrammatically. In euchromatin, what happens, children? This is a bead, then again here is the bead, here is a bead, here is a bead. That means the beaded structure, that means the nucleosome. जो nucleosome है उनमें distance होता है. They are no tightly packed, no high, there is no high coiling of DNA. लेकिन heterochromatin में क्या होता है? The nucleosomes are present like this. Okay, so that is called heterochromatin region. तो इन द सेम क्रोमोसोम में, एक ही क्रोमोसोम में डीएनए का यूक्रोमाटिक पार्ट होता है एंड हेटेरोक्रोमाटिक पार्ट भी होता है. तो ये दोनों में डिफरेंस, ये स्ट्रक्चरल डिफरेंस है बच्चों. दिस इज नॉट हाईली कॉइल्ड, नॉर्मली कॉइल्ड, बट इट इज हाईली कॉइल्ड पार्ट ऑफ द डीएनए. लेकिन इसका एडवांटेज एंड डिसएडवांटेज क्या है? ओके, सो हियर लेट मी राइट डाउन द पॉइंट्स. दोनों में डिफरेंस में लिख देती हूँ. In euchromatin is that region of chromatin where the region of chromatin where DNA is coiled loosely, but here DNA is coiled tightly. DNA is coiled tightly. Okay. Next, since here it is coiled loosely, so it is transcriptionally एक्टिव. ये ट्रांसक्रिप्शन कर सकता है, यानी डीएनए से आरएनए फॉर्मेशन हो सकता है. आरएनए बन सकता है, लेकिन दिस इज ट्रांसक्रिप्शनली इनएक्टिव. ये ट्रांसक्रिप्शनली इनएक्टिव है. और ये क्रोमोसोम के कौन से हिस्से में होता है? इट इज प्रेजेंट इन द आर्म्स ऑफ द क्रोमोसोम. But this is present in the centromere, telomere, जहां पर आपका आरएनए ट्रांसक्रिप्शन नहीं होता. ओके, एंड सिंस इट इज लूजली कॉइल्ड, सो इट इज लाइटली स्टेन्ड, but it is darkly stained. This is one more difference. So these are the differences between euchromatin, heterochromatin. So children, with this our today's session finishes, and today we have discussed the contribution of different scientists regarding the composition and structure of DNA, and also we discussed the packaging of DNA, and we discussed about euchromatin and heterochromatin, and I hope all the concepts are clear to you, and thank you very much. Thank you so much.